For centuries, the story of Britain has been told as a tapestry of kingdoms, invasions, and cultural myth. England, Scotland, Wales, each framed as a separate lineage, a set of static identities inherited from heroic or noble forebears. But the genetic record tells a different story. In many regions of the British Isles, the DNA of today's inhabitants reveals waves of migration, blending, and survival that contradict the neat categories of history books. Populations assumed to be Celtic or Anglo-Saxon show extensive admixture. What we have called heritage is often an artifact of selective memory, not biological isolation. Modern genome sequencing across the United Kingdom demonstrates that temporal continuity exists, but not in the form of unbroken cultural purity. Ancient DNA from Bronze Age burials in Wessex overlaps significantly with contemporary populations in southern England. Yet there is evidence of gene flow from continental Europe, reflecting repeated migrations over millennia. In contrast, samples from Western Wales and the Scottish Highlands suggest pockets of relative isolation, influenced by topography and settlement patterns, but these regions are far from genetically static. Drift and founder effects created measurable distinctiveness, but nothing resembling the myth of untouched ancestry. The first pivot becomes unavoidable when we examine ancient DNA from Britain alongside continental samples. During the Iron Age, the influx of Celtic-speaking populations from Gaul left detectable genomic signatures, but these signals were layered atop an already diverse genetic substrate. Cultural assimilation cannot be equated with biological replacement. By the early medieval period, Anglo-Saxon settlements contributed substantially to the English gene pool, yet there was no wholesale replacement. Ancestry remained a mosaic. The long-standing assumption that modern English identity derives predominantly from Anglo-Saxon invaders collapses under scrutiny. At the level of individual genomes, the picture grows more intricate. Runs of homozygosity, which indicate periods of relative isolation, are present in certain rural valleys and islands, yet these same populations also carry alleles introduced from long-distance migration. Bottlenecks occur, but they are punctuated by admixture, creating statistical distinctiveness without implying purity. Every simplification, every cultural myth about static peoples is undermined by this measurable complexity. The macro-level consequences of these findings is subtle but profound. The nations of Britain, often portrayed as emblematic of discrete cultural heritages, are instead microcosms of a universal human pattern: migration, admixture, adaptation. Civilization built fortifications, kingdoms, and borders, yet DNA flows across them, indifferent to narratives of origin. What appears as cultural identity is often the product of memory and myth. What persists biologically is continuity within change. Genetics does not honor historical pride or class distinctions. It registers survival, demographic shifts, and adaptation to both environment and circumstance. In Britain, the data forces a re-evaluation of assumptions about lineage. The familiar stories of pure Celts or Anglo-Saxons are not biologically supported. Instead, what we observe is a layered human record shaped by repeated population fluxes, topographical barriers, and selective pressures over thousands of years. The earliest layer of modern British genomes begins in the Mesolithic, roughly 10,000 years ago, when post-glacial hunter-gatherers spread across the islands. Analysis of skeletal remains from sites in southern England and the Scottish lowlands show small, mobile populations with genetic profiles closely related to Western European foragers. These groups carried alleles for dark hair and skin, traits gradually replaced or diluted by incoming farmers, but their legacy persists in subtle frequencies within the modern gene pool. Their legacy is continuity, not stasis. fragments of survival embedded in a dynamic genome. By 6,000 years ago, the Neolithic revolution introduced farmers from continental Europe. Genomes from early Neolithic burials in the Thames Valley demonstrate a substantial admixture of Anatolian-derived lineages with the local Mesolithic substrate. These farmers introduced not only new technologies, but new genetic variation, establishing a pattern that would repeat across the British Isles. Incoming populations merged with resident groups rather than replacing them. Isolated pockets persisted. Orkney and parts of Western Wales exhibit higher retention of Mesolithic ancestry, but no region remained untouched. The assumption of a purely indigenous population is therefore untenable. The Iron Age, beginning around 4,500 years ago, brought another wave of significant genetic change. Genome sequencing from Wessex burials reveals an influx associated with steppe-derived lineages. Y chromosome haplogroup RB1 expands dramatically in this period, while mitochondrial diversity remains high. These data indicate predominantly male-driven migration, likely linked to technological or social innovations rather than population replacement. Across Britain, this admixture reinforced the mosaic pattern, continuity of maternal lineages coupled with male-biased demographic shifts. Cultural labels like Celts are tempting, but genetics reveals no tidy correspondence. Ancestry is overlapping, not categorical. The pivot point becomes particularly clear during the Iron Age. DNA extracted from southern Britain reveals continued continental interaction, primarily with Gaul, but also sporadic inputs from other Atlantic populations. Linguistic evidence might suggest a uniform Celtic identity, yet genetic data shows that cultural homogenization rarely aligns with genetic uniformity. Admixture proportions vary locally. Coastal regions show higher continental contributions. Interior valleys retain more ancient substrate. These distinctions are statistical, not qualitative. They represent probabilities of shared alleles, not markers of authentic heritage. With the Anglo-Saxon migrations beginning around the 5th century CE, previous assumptions of genetic transformation come under scrutiny. High-resolution analysis of cemetery sites across East Anglia and Mercia reveals a range of admixture. Some individuals exhibit almost complete continental Germanic ancestry, while others remain heavily aligned with prior Bronze and Iron Age populations. The variance is significant, demonstrating that the demographic impact of the Anglo-Saxons was uneven, both geographically and socially. England today carries this layered signal, a palimpsest of population flux, rather than a simple inheritance from invaders. Scotland and Wales present contrasting microhistories. Highlands and islands show longer runs of homozygosity, consistent with relative isolation imposed by topography. Yet even here, DNA evidence indicates contact with Norse settlers, particularly in Orkney, Shetland, and the Western Isles. Wales exhibits a similar pattern. Valleys and upland regions retained older lineages, but migration through river corridors and coastal trade brought substantial genetic exchange. In both cases, isolation did not equal purity. Temporal continuity existed, but within a framework of continual genetic layering and adaptation. Genomic studies also reveal founder effects in localized populations. Certain alleles associated with disease susceptibility or metabolic adaptation are overrepresented in specific regions, reflecting historical bottlenecks. These effects are measurable and statistically significant, yet they do not correspond to cultural boundaries. An English village, a Highland glen, or a coastal Welsh settlement might carry distinctive allele frequencies, but the differences are quantitative, not qualitative. Myths of ancient, unbroken bloodlines collapse under the weight of empirical data. The interplay of drift, migration, and admixture shapes more than lineage. It influences adaptability. Alleles introduced from continental Europe brought new immune responses, metabolic traits, and phenotypic variation. The combination of continuity and admixture created resilience, enabling populations to survive environmental challenges, disease outbreaks, and social upheaval. Genetics demonstrates survival as a process, not a narrative. Identity is maintained through functional continuity, not static preservation. The first major recalibration arises when comparing modern genomes to these ancient baselines. A contemporary inhabitant of Cornwall may carry as much steppe-derived ancestry as a Londoner, yet local allele distributions reflect centuries of geographic and social constraints. Regional identity, as experienced culturally, rarely mirrors genetic reality. Continuity exists in probability, in allele frequencies, in subtle drift, not in the simplified heritage categories often deployed in popular discourse. By the early medieval period, the British Isles had become a laboratory of continuous human movement, adaptation, and genetic layering. The mosaic established during the Bronze and Iron Ages persisted, yet now it intertwined with social transformations. Kingdoms emerged, borders shifted, and new cultural identities were forged. Genetics shows that these changes occurred on a substrate of continuity. The alleles inherited from Mesolithic hunter-gatherers, Neolithic farmers, and Bronze Age migrants remained present, circulating quietly beneath the surface of written history. What appears as national identity in chronicles and myths is largely a cultural overlay imposed upon a living genetic record. The Norman conquest of 1066 provides a striking case study. Historical narratives describe wholesale replacement or domination, yet genomic analysis tells a more nuanced story. DNA from 11th and 12th century cemeteries in southern England shows that Norman settlers contributed to the gene pool, particularly through male lineages, but most of the population retained pre-existing genetic continuity. This aligns with patterns observed across Europe. Conquest rarely produces biological erasure. Instead, social hierarchies emerge, while the underlying population remains resilient, integrating newcomers selectively. Temporal continuity and admixture coexist, a recurring theme across millennia. Regional variation further illustrates the complexity of identity. In Scotland, the highlands and islands remained partially insulated by rugged terrain, yet Y chromosome data reveals Norse influence in Orkney and Shetland, exceeding 25% of paternal lineages. Maternal lineages show less perturbation, demonstrating gender-biased migration patterns consistent with Viking settlement strategies. In Wales exhibits analogous dynamics, with western valleys preserving higher frequencies of Iron Age haplogroups, while eastern lowlands reflect Anglo-Norman admixture. Across Britain, topography, trade routes, and conquest dictated the degree of genetic continuity, creating statistical distinctiveness without implying cultural or racial purity. The concept of cultural memory complicates interpretation. Myths of Celtic purity, Anglo-Saxon dominance, or Viking conquest emerge because social narratives selectively emphasize events that matter culturally, not biologically. Genetics provides a corrective lens. Each wave of migration, Mesolithic, Neolithic, Bronze Age, Iron Age, Anglo-Saxon, Norse, Norman, layered alleles onto existing populations. The measurable outcomes are probabilities of shared ancestry, allele frequencies, and drift signatures. What survives as cultural identity is only a shadow of this deeper biological reality. Yet even within this framework, adaptation is evident. Certain regions of Britain exhibit signatures of positive selection. Alleles related to lactase persistence rose during the Neolithic and Bronze Age, conferring dietary flexibility. Immune-related alleles introduced via continental gene flow mitigated vulnerability to pathogens introduced through trade and migration. These traits illustrate a critical point. Genetic continuity is not about stasis, it is about survival. Populations persist because they adapt, incorporate novelty, and maintain functional resilience. Continuity is measurable, but it is adaptive rather than nostalgic. The second pivot emerges in the analysis of urban versus rural populations. Industrialization, starting in the 18th century, reshaped genetic landscapes in ways that classical historical narratives rarely capture. Massive internal migration from the Welsh valleys, Scottish highlands, and northern English coalfields to growing urban centers like London, Manchester, and Glasgow redistributed alleles across previously semi-isolated regions. Patterns of admixture accelerated, founder effects diminished, and statistical distinctions between regions blurred. The genome of the English city became a microcosm of the long-term British pattern: layered ancestry, admixture, and adaptive continuity. Consider the English Midlands. Genomic-wide analyses reveal that the influx of laborers from rural counties during the Industrial Revolution increased haplotype diversity, while maternal lineages from isolated villages were preserved in small pockets. Cultural memory might remember the migrants' origins in terms of dialect or occupation, but their DNA records a subtler narrative: movement, integration, and survival. The broader implication is clear. Identity, as recorded in genetics, is probabilistic, not categorical. It records what survived demographically and what adapted functionally, not what history or folklore celebrates. Even at the macro scale of national identity, these patterns hold. Modern genetic clustering across England, Scotland, and Wales shows overlaps that defy political or cultural boundaries. Southern England exhibits higher continental admixture, but no region is free from the layered histories of Mesolithic, Neolithic, and Bronze Age ancestry. The Highlands and Welsh valleys retain subtle statistical distinctions, but they are gradients rather than absolute separations. Continuity exists, but always within a framework of flux. This challenges popular assumptions that cultural or national identity corresponds to clear-cut genetic categories. The long-term evolutionary perspective offers a final insight. Across 10,000 years, the British genome has absorbed and retained variation through cycles of migration, admixture, and selective pressures. Distinctiveness appears in specific alleles, regional haplogroups, or patterns of homozygosity, but there is no genetic equivalent to historical myths of purity or cultural stasis. Instead, what persists is a record of resilience, populations that survived environmental stress, social upheaval, and demographic bottlenecks, not because they were untouched, but because they incorporated new variation without losing continuity. The map of humanity wasn't written in a lab. It was carved by the sea and the stone. This is Helix Horizon, where we trace the code that refuses to stay still. In contemporary Britain, the echoes of ancient migrations remain embedded in genomes, yet they rarely align with perceptions of ancestry or class. Modern Londoners, for example, carry alleles tracing back to Bronze Age farmers, Anglo-Saxons, and Norse settlers, sometimes within a single chromosome. These signals exist beneath the surface of social labels, occupational histories, and regional pride. Genetics demonstrates that continuity is probabilistic. The persistence of ancestral lineages does not correspond to cultural or socioeconomic identity, but to survival and demographic integration over thousands of years. Population-level studies reveal subtle regional structure that mirrors geography more than mythology. Coastal and riverine corridors show higher continental admixture, reflecting historical trade and migration. whereas inland valleys preserve alleles indicative of relative isolation. Yet even in areas once considered genetically remote, admixture is pervasive. Cultural narratives that elevate Celtic heritage, Anglo-Saxon purity, or Viking dominance obscure a far more complex reality. Identity is a product of layered ancestry, mobility, and adaptation. Industrial and post-industrial migration further complicates the picture. From the 18th century onward, rural populations relocated en masse to urban centers, mixing previously semi-isolated lineages. Genome-wide association studies demonstrate that these migrations reduced local founder effects and increased heterozygosity in metropolitan populations. Contemporary genetic variation in cities like Manchester or Glasgow is therefore not a reflection of any single ancestral group, but the accumulated result of millennia of movement, admixture, and selective pressures. The historical impact of class and occupation is visible socially, yet largely invisible genetically. The same principle applies when examining mobility across social strata. Occupational inheritance and regional settlement patterns created apparent continuity within social classes, yet DNA reveals no corresponding biological barrier. Alleles traverse class boundaries through marriage, migration, and demographic chance. Claims of genetic distinction between elite and common ancestry are unsupported. Measurable differentiation exists only in statistical gradients, not categorical separation. These patterns illustrate a central evolutionary insight. Survival and continuity are shaped by opportunity and adaptation, not social myth. This insight recalibrates our understanding of British identity itself. While historical memory emphasizes kingdoms, dialects, and territorial claims, genetic evidence reveals a much more fluid map. English, Scottish, and Welsh populations share a deep continuity that predates recorded history, yet each region carries signatures of repeated migration and selective pressures. Identity, as experienced culturally, is a reflection of memory and social structure, whereas ancestry, as recorded biologically, is a probabilistic record of survival across millennia. Urbanization and modern mobility continue this process today. Genetic studies demonstrate that interregional admixture in the UK has accelerated over the past century, creating populations in which no single ancestral group dominates. Yet subtle statistical distinctiveness persists in some rural areas, demonstrating that geography and social structure continue to shape lineage distributions. Evolutionary forces operate alongside culture, yet in ways invisible to historical perception. Survival, adaptation, and integration remain the constants. Even within culturally celebrated lineages, genetic analysis frequently reveals surprising admixture. Families long thought to descend from a singular regional or noble origin often carry alleles tracing back to distant populations, reflecting centuries of migration and gene flow. Continuity is not about isolation. It is about the layering of variation across generations. These insights challenge romanticized assumptions about heritage, demonstrating that biological reality is far more nuanced than cultural narrative. The broader evolutionary lesson is equally clear. Human populations persist through adaptation, not stasis. Genetic continuity is measurable across time, but it is always conditioned by migration, admixture, and environmental pressures. The British Isles exemplify this dynamic. Alleles introduced by Mesolithic foragers, Neolithic farmers, Bronze Age migrants, and successive waves of continental settlers have survived in complex patterns, producing populations that are resilient, adaptable, and genetically layered. Heritage is continuity in motion, not an unbroken chain of purity. As the evidence accumulates, one final recalibration emerges. Our understanding of identity and ancestry must integrate both biology and history without conflating the two.
Genetics cannot tell us who we are culturally, but it can reveal how populations have persisted, mixed, and adapted. The persistence of ancient alleles alongside recent admixture illustrates a fundamental principle. Survival is a dynamic process, not a static inheritance. The genomes of the British inhabitants are living archives of this principle. Yet even as we map these patterns, the story is incomplete. Ancient DNA, while illuminating, represents snapshots, often fragmentary, of a far larger and more complex demographic history. Many migration events remain undersampled. Local founder effects are only partially understood. Selective pressures are inferred but not fully reconstructed. Genetics provides unprecedented clarity, yet it also underscores the limits of certainty. Human continuity is measurable, but never fully predictable.
Across the British Isles, the genome is neither tidy nor static. It carries the imprints of countless migrations, partial isolations, and selective pressures. Yet these signals are often subtle, dispersed, and probabilistic. The alleles that endure are those that facilitated survival, a mosaic shaped by environment, demography, and chance, rather than by conscious cultural choices. Ancient myths of Celtic purity, Anglo-Saxon dominance, or Viking conquest collapse under genomic scrutiny, revealing instead a record of resilience in motion. Even in regions traditionally considered isolated, subtle admixture signals persist. Highland valleys, Welsh uplands, and Cornish coasts all show evidence of repeated gene flow over millennia. Isolation does not equal stagnation. Drift produces measurable distinctiveness, but never biological absolutism. Temporal continuity coexists with layered variation, producing populations that are distinctive in statistical terms, yet wholly integrated within the broader human pattern. Genetics demonstrates that human persistence is not a matter of purity, but of survival, adaptation, and flexibility. The implications extend beyond the British Isles. Across every continent, every population, human history has been a series of waves: migration, admixture, adaptation, and partial isolation. What we observe in Britain is a microcosm of a universal process. Civilization imposes borders, cultural memory imposes narratives, yet biology follows its own logic. Continuity emerges not from stasis, but from repeated layering, the survival of traits and lineages across time and space. Modern genomics allows us to detect even faint echoes of these dynamics. Allele frequency shifts, runs of homozygosity, Y chromosome haplogroup expansions, mitochondrial persistence, all signal a complex balance of drift, admixture, and selection. Yet the record is incomplete. Many regions remain undersampled. Temporal gaps persist. Environmental and social pressures interact in ways that are difficult to reconstruct fully. The story of human survival is always partially hidden, waiting for new data to illuminate its next layer. This partial visibility is itself instructive. It demonstrates the limits of cultural memory. The identities we celebrate, the histories we canonize, often obscure the deeper, measurable continuity embedded in DNA. Genetics neither confirms nor invalidates cultural narratives. It reframes them. A Highlander may carry Norse alleles. A Londoner may retain Mesolithic ancestry. Social labels capture meaning, but biology captures survival. Continuity is statistical, functional, and layered, never absolute, never static. And yet questions remain. How did local selective pressure shape the distribution of rare alleles in specific valleys or islands? To what extent did repeated migrations interact with climate fluctuations to influence adaptation? How much of the genetic legacy is invisible to current sampling, waiting in unexcavated burials or underrepresented rural populations? Every genome is a partial record, every population a living archive of incomplete knowledge. As we look forward, the British Isles serve as both case study and metaphor. Human continuity is always a palimpsest. Survival over millennia is written in fragments, layered by migration, admixture, and adaptation. The patterns we observe today are the result of processes ongoing since the first post-glacial settlers stepped onto these islands, processes that continue in every city, valley, and coast. Understanding them requires patience, precision, and humility. The unanswered questions beckon. What hidden signals lie in unsampled regions of Britain? How did alleles introduced thousands of years ago interact with modern selective pressures from industrial pollution to urbanization? And across the globe, how do similar patterns of continuity and admixture inform our understanding of human resilience as a species? Each answer uncovers new complexities, new layers of history, new evidence of survival in motion. The map of human ancestry, already intricate and unresolved, is far from complete. Britain's layered genomes are one chapter in a story that stretches across continents and millennia, a story written in survival rather than myth, in adaptation rather than static identity. The door remains open, inviting deeper exploration into the forces that shape humanity's continuity. Forces recorded in DNA, yet only partially visible, waiting for the next discovery to illuminate what we have not yet seen.